Good morning everyone. I am Yun. Thank you for being here for my research presentation. My topic is in silico genetic variation analysis of cytochrome P452C19 and their effect in certain drugs metabolism.
Your topic sounds interesting, Miss Yoon. Let me know more, please.
Thank you, Sam. Let's get into my research.
In silicon is a computational approach to evaluate the potential toxicological risk of a drug candidate. The computational approaches can be such as polyphenol 2, NCBI, FARM-MGKB, and many others. In silicon is important due to the presence of genes polymorphism. The question is, what is polymorphism? Polymorphism is a difference in DNA sequence among individuals, groups, or population which can be arise from mutation. While these differences in DNA has been associated with diseases. Today, I'm going to focus one of the polymorphisms that occur in cytochrome 2C19. This cytochrome 2C19 is located at chromosome 10, which is responsible for at least 10% of commonly prescribed drugs, metabolism, and processing. Therefore, if there is polymorphism in this gene, most impacts on the metabolism of therapeutic drugs can be seen. There are about 28 variants that have been identified in this cytochrome 2C19. All these variants can cause variations in the pharmacokinetics and pharmacodynamics, as well as associated with adverse drug reaction or therapeutic failure. Here, I provided some of the examples of therapeutic drugs which have been affected by drug metabolism cytochrome 2C19. As we know, single nucleotide polymorphism SNP can determine the risk of an individual's susceptibility to many illnesses and their drug responses, while non-synonymous SNP and SSNP are one of coding SNP types, leading to the diversity of encoded human proteins, which able to affect gene regulation by altering DNA and transcriptional binding factors, maintain the structural integrity of the cells, as well as affect protein's function. Therefore, by identifying and analyzing NSSNP can help in understanding their effects on genes product and their association with diseases, as well as development of new medical testing markers and individualized medication treatment. In this study, I used several bioinformatics software to investigate the effect of SNP on protein structure and function, as well as to discover whether genetic variation can affect dosage efficacy, toxicity, and pharmacokinetic activity. Then, here come to my methodology.
The first method that I have used in my research is by searching datasets. So, actually all of the information regarding the SNP found in human cytochrome 2C19 gene were retrieved using SNP database. It is actually a genetic variation database that established by the National Center of Biotechnology Information (NCBI). All of the retrieved data included the protein accession number of cytochrome 2C19, such as all this number, and all the SNP IDs that have found in this gene. The second method that I have used is SIFT, sorting tolerance from intolerance. It is actually an online bioinformatics software that uses an algorithm to predict the effects of amino acid substitutions. It uses the sequence homology to predict the effects of all possible substitution at each position in the protein sequence. So this is the software that I have used, and I found it very interesting and very useful. I use a software named Polymorphism Phenotyping V2, or we call it as PolyPhin2, to predict the functional modification or the damaging effects of missense mutation of coding NSSNP, which is non-synonymous SNP. The non-synonymous SNP that were predicted to be deleterious by SIFT were submitted to this software, and then the prediction result of two software will then be compared with each other and pick out those SNP who were in common, meaning those SNP that has double positive results in two software. The very last method that I used was using PharmaGKB. It is the first genomic web platform that provides the opportunities to evaluate the genetic differences among populations for all autosomal genes. PGKB also serves as an integrated platform for linking this data with drugs and genetic variants that affects drug responses, adverse reaction, and dosage requirement. So in my research, what I have need to do is just by inserting my SNP into this software, and then the software will evaluate the SNP on the drug response.
From my study, I found out that cytochrome 2C19 is very important causative factor for drug interaction with clinical diseases such as coronary artery disease, peptic ulcer disease, Helicobacter pylori infection, and others. Scan this QR code to obtain more information regarding to the result. In conclusion, the use of in silico tools for application in biomedical research is highly effective and has a great impact on the ability to discover the genetic mutations that cause variation in dosage efficacy, toxicity, and pharmacokinetic.
It's such an amazing presentation, Miss Yoon.
Really? Thank you very much. I am really happy to hear this. Hope that my sharing could bring you some input.
Sure, I got new knowledge for me to bring home. See you again in coming conference next week.
See you next week, Sam.